Hello students, today we would be doing spermatogenesis from the chapter Human Reproduction. If you haven't seen the previous part, I suggest you see the previous part which deals with the male and female reproductive system and then continue with this part. Gametogenesis. As we have discussed previously, gametogenesis. Genesis means formation, gametogenesis is formation of gametes. The process of formation of haploid gametes from the undifferentiated diploid germ cells in the gonads is called as gametogenesis. So here we are getting haploid gametes, that is, suppose the animal has 2n number of chromosomes, the gamete will be having just half of it, that is n number of chromosomes. So gametogenesis in case of humans we can divide into two, that is one in male and the other is in female. In male it is called as spermatogenesis and in female it is called as oogenesis. Spermatogenesis occurs in testes where sperms are produced. Oogenesis occurs in ovary where ova is produced. Gametogenesis is divided into three phases. One, the multiplication phase where the germ cells divide by mitosis, that is equational division and increase in number. Two, growth phase where these germ cells which have increased in number, they start growing in size. 3. Maturation phase where the cells undergo meiosis, that is reductional division to produce the gametes. Spermatogenesis. Where does it occur? It occurs in seminiferous tubules of testes. These seminiferous tubules are lined by epithelium which is called as germinal epithelium. Germinal epithelium, it contains the spermatogonial cells and the Sertoli cells. When does the spermatogenesis occur? It occurs after puberty. So, puberty is a period when the reproductive organs become functional. It is attained at the age of 13 to 16 years. It is triggered by the secretion of hormone testosterone in the testes. So, spermatogenesis starts because of the secretion of the hormone testosterone. This hormone brings about growth and maturation of secondary sex organs and development of accessory sex characters. So what are the secondary sex organs? The primary sex organ would be the testes, while the other parts like the seminiferous tubules, epididymis, sperm ducts, seminal vesicles, prostate glands, corpus glands would be the secondary sex organs. And what are the secondary sex characteristics? That is growth of body and facial hair, enlargement of larynx, broadening of shoulders, are all the secondary sex characteristics in case of males. Spermatogenesis can be divided into two parts. One, the formation of spermatids, and the second is formation of spermatozoa. Spermatozoa is nothing but the sperm. The formation of spermatids, it can be divided into three phases. One is multiplication phase, growth phase, and the maturation phase. And this spermatogenesis, that is, spermatogenesis has both the formation of spermatids as well as the spermatozoa. But this process where after maturation, where these four haploid spermatids are formed, they are not completely functional sperms. So these sperms, by the process of spermiogenesis, they give rise to the 4 haploid spermatozoa or the sperms, which are the functional sperms. So this process where the 4 haploid spermatids are transformed into the spermatozoa or the sperm is called as spermiogenesis. Spermatogenesis, let us see in a little more detailed manner. In spermatogenesis, as we have been seeing, gametogenesis is divided into 3 phases. So here, spermatogenesis, again, we will study in 3 phases. One is multiplication phase, growth phase, and maturation phase. Multiplication phase, the spermatogonial cells, which are 2n in number, that is, it is diploid. The spermatogonial cells are diploid. They divide mitotically to give rise to more number of cells, right? In multiplication phase, the spermatogonial cells, they divide mitotically to give rise to more number of cells. These spermatogonial cells, among these spermatogonial cells, some of the spermatogonial cells, some of them, they stop dividing and they enter into the growth phase. They start growing in size. And when they grow in size, they accumulate a lot of cytoplasm and they're ready for further process. So after the spermatogonial cell grows, it is then called as primary spermatocytes. Since primary spermatocyte is formed in this phase, which is the growth phase, this phase is also called as spermatocytogenesis, that is formation of spermatocyte. Growth phase is also called as spermatocytogenesis. In the maturation phase, this primary spermatocyte, which is still a diploid cell, undergoes meiosis 1, which is a reductional division to give a haploid secondary spermatocyte. The secondary spermatocyte undergoes a second division to give rise to the 4 spermatids. So the second meiotic division is a reductional division, right? So we'll go through the slide once again. Spermatogenesis is divided into 3 processes. One is the multiplication phase, the growth phase, and maturation phase. In the multiplication phase, the spermatogonial cells, which are diploid cells, undergo mitosis and increase in number. Some of these spermatogonial cells stop dividing and they start growing in size. After they grow to a certain size, they go to the next phase, which is maturation phase. Here, the primary spermatocyte, which is still diploid, undergoes first meiotic division, which is a reductional division to form the secondary spermatocyte, which is now haploid cell. The secondary spermatocyte undergoes a second meiotic division, which is an equational division. Here, the number of chromosomes is not going to. So this is our NCERT diagram. So we are seeing here the spermatogonia. It is undergoing mitosis. It is equational division and it is forming many cells. So some of the cells, that is, here we can see one spermatogonium which will grow in size, and this forms the primary spermatocyte. This primary spermatocyte undergoes meiotic division and it forms the cells which are half in number. So after first meiotic division, which is a reductional division, we have the secondary spermatocytes which are haploid in number. So the chromosome number, which was 46, has now become into 23. Now the secondary spermatocyte undergoes the second meiotic division, which is an equational division. So here you see the spermatids are still 23. That is, the spermatids still have 23 number of chromosomes. So it has n number of chromosomes and the spermatogonial cell have 2n number of chromosomes. The spermatids undergo differentiation to form the spermatozoa. This is another diagram which is easy to practice. I would suggest you learn this diagram for your examination. Here again, the spermatogenesis is divided into 3 phases, which is multiplication phase, growth phase, and maturation phase. And the differentiation phase, that is your spermatogenesis, is shown over here at the last. So here you have the primary germ cell, which is having 2n number of chromosomes. They will divide and form the spermatogonial cells, which also have 2n chromosomes. These spermatogonial cells, some of these spermatogonial cells will go to the growth phase, that is, they start growing in size and form the primary spermatocyte, which is still having 2n amount of chromosomes. The primary spermatocyte undergoes meiosis, which is a reductional division, and it forms the secondary spermatocytes, which is having half the number of chromosomes. These secondary spermatocytes undergo a second meiotic division, which is an equational division, to form the spermatids. These spermatids undergo the process of spermiogenesis and differentiate into sperms. So here is a diagram. You can see this is the testis, and in the testis the sperms are formed. Spermatogenesis occurs in the seminiferous tubules. If you take a section of the seminiferous tubule, this is what it would look like, the structure of which we have already discussed previously in the previous part. If you take a section of this part, the section of this is how it looks. This is the Sertoli cells, which is the nursing cells, and then you have different types of these spermatocytes here. You have the primary spermatocyte, the secondary spermatocytes, which form the spermatids, and the spermatozoa. This is a textbook diagram. Here you have the germinal epithelial layer. This is the germinal epithelial layer, outer layer. Inner to that you have the spermatogonial cells. These are your spermatogonial cells. And you have Sertoli cells also, which we have discussed previously. And then you have the primary spermatocyte, which is formed over here. And then you have the secondary spermatocyte. And here you can see the spermatid is still attached to the Sertoli cells. And when the Sertoli cell, spermatid is released from the Sertoli cells, you have the formation of the spermatozoa. This is a histological section of your seminiferous tubule. So here you can see the Sertoli cells. These are the spermatozoa, and these cells are the spermatocytes. Another picture for you to appreciate the same amount of cells so that you understand it more clearly. Now, spermiation. This is another term that we have understood. The concept we have already understood in the previous slides. So, spermiation is the process by which spermatids are released from the Sertoli into the seminiferous tubules. So, I just told you the spermatids are still attached to the Sertoli cells, that is the feeding cells or the nurse cells, and the spermatids have to release themselves from the Sertoli cells. So after spermiogenesis, after the formation of these spermatids, the sperms, they release themselves from the Sertoli cells, and this process is called as spermiation. So this spermatozoa or the sperms which are released, they enter into this region, which is the seminiferous tubules lumen. So sperms released from the seminiferous tubules are transported by the accessory ducts. What are the accessory ducts? The rete testis, vasa efferentia, epididymis, vas deferens. These are all the accessory ducts. The secretion of the epididymis, vas deferens, seminal vesicle and prostate, they all help in maturation and motility of sperms. The seminal plasma, along with the sperms, constitute the semen. Now we have already seen the function of the male sex accessory ducts and glands are maintained by the testicular hormone, which is the androgen. Let us now understand the hormonal control of spermatogenesis. Gonadotropic releasing hormone is a hormone which is released from the hypothalamus. That is, it is a hypothalamic hormone. This acts on the anterior pituitary. So, in the 11th standard we have studied pituitary has the posterior part and the anterior part. So, the anterior pituitary releases the gonadotropins. Gonadotropins, which are the luteinizing hormone and the follicle stimulating hormone. Luteinizing hormone, it acts on the Leydig cells. These Leydig cells, they synthesize and secrete androgens. Mostly here we are talking basically about the testosterone, which stimulates the spermatogenesis. So androgen, that is testosterone, it stimulates spermatogenesis, the process which we have already studied in the previous slide. Now coming to follicle stimulating hormone, this acts on the Sertoli cells. Sertoli cells are the nerve cells. It secretes factors which help in spermiogenesis. What is spermiogenesis? That is formation of the spermatozoa. How is the level of testosterone maintained within the body? So we have understood the hormonal regulation where the gonadotropins are released from the anterior pituitary. Those are the luteinizing hormone and follicle stimulating hormone. The luteinizing hormone will act on Leydig cells to synthesize the androgen, which is testosterone. And here the follicle stimulating hormone, it acts on the Sertoli cells, which helps in spermiogenesis. The Sertoli cells, along with secreting these factors which help in spermiogenesis, it also secretes the inhibin, which will inhibit the synthesis of follicle stimulating hormone. Similarly, the testosterone, the excess of testosterone, will inhibit the luteinizing hormone. Thus, the hormonal synthesis is regulated. Now, let us see the structure of sperm. Sperm is a microscopic structure which is about 50 micrometers in length. It is composed of a head, neck, and middle piece and a tail. The sperm is enveloped by a plasma membrane. So if you see the sperm, all over you have a membrane which is the plasma membrane. The sperm head contains an elongated haploid nucleus. So this is your nucleus which is elongated and it is haploid. So then the chromosome number is half than the somatic cells. And it is covered by a cap-like structure which is called the acrosome. Acrosome has enzymes which are needed for fertilization. The middle piece, it has a lot of mitochondria. You can see a lot of mitochondria are there in the middle piece. These mitochondria, we know, are the sites of synthesis of ATP. So they produce energy to help the sperm to move, which is essential for fertilization to reach the ova. Till now, we have understood the whole process of spermatogenesis. Let us now see what is the difference between spermatogenesis and spermiogenesis. Spermatogenesis is the process of formation of haploid spermatozoa from undifferentiated diploid spermatogonial cells, whereas the spermiogenesis is the process of transformation of spermatids into spermatozoa. Here the spermatid change into spermatozoa. Spermatogenesis involves multiplication phase, growth phase, and maturation phase, whereas spermiogenesis is a part of spermatogenesis that includes only the maturation phase. Here, one spermatogonium gives rise to 4 spermatozoa, but in the spermiogenesis, one spermatid, that is, one spermatid gives rise to 1 spermatozoa. In the next part, we will be learning about oogenesis. Thank you for now.